Hi guys, can you hear me? I was just having some problem with my microphone, but I think it might be sorted now. Just send me a message if you can all hear me. Okay, nobody sent me any messages. I don't know if you can hear me or not. Can one of you just send me a message quickly just to say if you can hear me? Okay, great, thank you. Okay, I'm just going to turn off the public chat and put the private chat on. Okay, great. So welcome back, everyone. I hope you had a good couple of weeks, and I hope revision has been going well. We've got sort of just around 4 weeks left, really, before the exam. So yeah, not to scare you, but I hope that things are going well and that you guys are kind of getting through the content, starting to do like lots of questions and stuff. So today we're going to go over molecular genetics. I do quite like this topic because I think that it is quite sort of straightforward to remember. If you just sort of learn it, you don't have to really understand a lot of the stuff. It's just kind of memorization again. So today we're going to go through all the molecular genetics stuff, pretty much completely syllabus, like covering everything really, because it's not really that massive. It's just considered a part of the previous section. So I think what we'll do is we'll start with the genetics of conditions, and then we'll go through and cover all the other things like cell cycle, and those kind of aspects as well. And if there's any questions, as usual, just put them in the chat, and I'll look at them at the end. But obviously if there's any problems or anything with the microphone or anything, just let me know and I will just have a look at that. So let's start off by looking at ADHD. So we're looking at the genetics of ADHD itself. So the heritability is around 74 to 85% and is similar in males and females. Interestingly, it's sort of not a single gene, which does make sense. It's got a very complex inheritance pattern, so multiple genes are really involved. And also there's other aspects that can be involved as well. So kind of other developmental disorders, other syndromes, other genetic disorders can have kind of predisposition to ADHD. And 2/3 of people who have ADHD continue to have symptoms in adulthood as well. And so some of those genes are listed there. So DAT1, DR, D4, DRD5. I'd say probably just knowing that it's highly heritable is probably the main point for ADHD, and that it's a mixed genetic inheritance rather than a single gene. Then moving on to alcoholism. So as you probably know, that biological children of parents who have alcohol misuse disorders are more likely to develop it even when they're adopted by parents who don't use alcohol or are not alcohol dependent. So it does show that there is a highly heritable aspect to alcohol misuse disorders. And as would expected in any heritable disease, the monozygotic twins has greater concordance than dizygotic. So that means that there's more understanding that it's heritable. Again, it's also complex genetic inheritance profile, but ADH1B and ALDH2 contribute to the risk. So those are probably the two to remember for alcohol. So ADH1B and ALDH2, and again, also highly heritable condition as well. So this is again related to alcohol. So this is the Asian flush reaction. So the enzyme, which is ALDH2504K, so anyone who has that would be displaying the Asian flushing reaction. And actually it has been known to be protective against misuse, and it has been theorized that maybe the reason why this enzyme kind of is has continued in evolution or like genetically is because it is basically protective against people becoming dependent on alcohol. So this gene, this enzyme that's encoded by a gene is quite common in Far East Asians, but also in other Asian populations as well. So one significant genetic polymorphism of the ALDH2 gene is present in 50% of Japanese, Han Chinese, and Taiwanese populations, and that's ALDH2 too. So basically, that's kind of, it's a protective kind of thing against misuse. And then these are the alleles. So we mentioned that before, but people who have the ADH1C and ADH1B2, they metabolize alcohol fast. And the most common one in Asians is the ADH1B2. And then the slow allele is common in Caucasians. So the 95% of Caucasian people are homozygous for the ADH1B1 slow allele. And that's also common in the African population as well. So yeah, maybe just make a note of that because those are the alleles that are relevant for alcohol misuse disorders. So looking at autism, again, this is another very highly heritable disorder. It's got a very strong genetic component. And again, with twins, it's sort of 60 to 90% of twin concordance. Siblings of autistic individuals are 50 to 100 times more likely to develop autism than the normal population. And there is also for autism complex genetic inheritance. There's not any specific particular one gene that is understood to be most specific for autism, but there are various candidate genes that have been suggested, and also something called copy number variations, which we'll cover later on in the presentation, but those kind of all are implicated in autism. And again, as you probably know, autism is also linked to sort of other neurodiversity. So if people are inheriting syndromes or any other kind of genetic disorders, they may be at increased risk of autism. And again, with all of these kind of autism, ADHD, there's a lot of crossover, and there's an increased risk of having other psychiatric comorbidities with those. They tend not to occur in isolation. So oftentimes people will have autism with anxiety or other things. And I'm sure you've all seen that in clinical practice as well. But that's why it's considered to have a complex genetic inheritance, because there's no specific one thing which makes someone have autism or not. So dyslexia, so this is a fundamental cognitive considered disability, but I think it's more commonly accepted now as just part of neurodiversity. Obviously, people with dyslexia can have normal levels of IQ or above average IQ. It's not necessarily related to like a learning disability as such, but it can be. Again, it's highly heritable, but there are some candidate genes that have been implicated for dyslexia. DCDC2 is probably the most common one that's cited, but the rest of those are also candidate genes. I wouldn't get too worried about learning all of those in particular, because this is quite like a specific thing, like dyslexia genetics, but maybe just remember DCDC2 for dyslexia. And that is highly heritable as well, and that they can also have normal and above average IQ. It is not necessarily implicated that they would have other learning disability or disorders. In fact, many doctors have dyslexia, and many professionals as well in other fields. Yeah, so basically having a look at the heritability, so Peterson in 2019 had a look at all of these, and basically ADHD, which we discussed at the start, is the most heritable, then followed by autism. Schizophrenia is again highly heritable, bipolar disorder, then anorexia, then alcohol dependence. I think alcohol dependence also has been the value changes in different studies, like because it can be hard to quantify what exactly alcohol dependence is clinically. There is an understanding that it is highly heritable, but there are various kind of numbers for how severe the heritability factor is. So I wouldn't necessarily say that it's only 35%, it can be higher than that. And then major depression and OCD. So it is definitely worth knowing those and seeing how those can come up in EMQs in the exam, sort of matching up what the heritability is like. So definitely just remember ADHD and autism at the top, and then OCD and depression sort of towards the bottom. But again, schizophrenia and bipolar are above 50% on average in terms of heritability. So schizophrenia we'll just touch on in a bit more depth because it is quite important. So yeah, looking at these candidate genes, schizophrenia is linked to a number of candidate genes, but no single gene is thought to be responsible. But it's thought that if there's more than one of these that has some kind of disorder, then obviously that will increase the risk of schizophrenia. Again, it's a complex inheritance profile, but the most important ones to note of those are neuregulin, dysbindin, DISC1, and COMT. So yeah, those are probably the most important ones to note. Yeah, I would say that. Also, I think DRD2 has come up before in a question. So, but yeah, DTNBP1, which is dysbindin, COMT, neuregulin, DISC1, and then maybe DRD2, probably just to make a note for schizophrenia. And we'll just touch on COMT a bit more. I don't even know if that's what people call it, COMT. That's what I call it. Maybe people are calling it by its full name, but yeah, I don't know who has time for that catechol O-methyltransferase. But basically, its main role is in breaking down dopamine, epinephrine, and norepinephrine, which are the catecholamines. And COMT, I think we should call it that, is the strongest schizophrenia candidate gene among the 27 genes that have been studied. They did a study looking at patients with DiGeorge syndrome, which is also known as velocardiofacial, and they have a lot of risk of psychosis. And they found that out of those 27 genes in a region on chromosome 22, the one that has the most sort of strongest risk factor for psychosis and in association to that schizophrenia is COMT. So that's probably the one to note. Yeah, and again, also for OCD as well. That's just what it looks like. And then neuregulin, that was the other main one. So they did a study on some Icelandic people who have like a family who had schizophrenia, and basically just found that this is relevant for risk factors. So the increased neuregulin signal suppresses the NMDA receptor, which lowers glutamate. This is not like 100% confirmed. It's just this is a theory that they've used to try and understand how it might work with the neuregulin, but I wouldn't worry about learning that exactly as it is. I think I would just remember that the name and that it's relevant for growth factor for neurons and that it's implicated in schizophrenia genetics. And then dysbindin was the other one, DTNBP1. I think I did get a question on this in the June paper last year. So this is found in muscle and brain tissue. And yeah, so the dysbindin expression is reduced in schizophrenia. And again, it's postulated that this is related to reduced glutamate as well. But dysbindin is normally part of the biogenesis, so the creation and formation of lysosome-related organelles. So that's a relevant enzyme that would be used. Yeah. And then DISC1, so this encodes a multifunctional protein which is involved in loads of different things. such as kind of creating the synapses between neurons, the structure of different neurons, the internal architecture, transport. So it's basically very got a lot of jobs, disc 1, and there's a Scottish family who had really severe kind of psychotic illness inheritance and schizophrenia, and they found they did a study and found that disc 1 disorder was really common. So that's the genetic understanding for schizophrenia. So just remember those ones. So they are disc 1, dysbinding, neuroglin, and COMT are your ones for schizophrenia. It can be a bit sort of difficult to remember because they're just a bit random names, but yeah, worth remembering those. And then the risk of developing schizophrenia. So you may have seen this kind of written out as a table. So I find it hard to sometimes to remember tables just like that. So going from the highest risk you can imagine is identical twins. So they've got 48% risk. Then what's called dual matings, which sounds a bit rude, kind of. It's basically saying that two people who are mates, who both have schizophrenia, the risk of the child having schizophrenia is then 46%. Fraternal twins, so 17%, and then sort of children of someone is 13%, full sibling is 9%, half siblings and parents 6%. So interestingly, so if a person has schizophrenia, there's a 6% chance that their parent would then develop schizophrenia, or I mean, it'd be weird if they had not had it first, but I suppose it could happen that way around. Grandchildren is 5%, nephew and niece is 4%, uncle, aunt, and first cousin is only 2%, and the lowest risk is just in the general population, which is 1%, which is still quite high, I think. So these are the relationships to a person with schizophrenia, and this is their lifetime risk of developing it. And adoption studies do show increased risk in biological relatives. There's incomplete penetrance, which we will cover again later, kind of what that means. But essentially, when you've got twins that are from the same zygote, so monozygotic twins, so identical twins, the concordance is around 50%, whereas in dizygotic twins, it's around 17%. That's written here as fraternal twins in the study. The heritability estimate is 80%, and segregation analysis suggests a multifactorial model. So again, lots of different kind of complex aspects to developing schizophrenia. So now we're going to go through the inherited disorders. I've pretty much included all of the ones that are kind of listed in any of the revision notes that you can find, and hopefully if you have an idea of most of these, then that should cover you for the exam. And there's kind of little ways I use to remember them, and I will just touch on those as well as we go along. So Rett syndrome, I think the key thing about this is it only affects females, so it only affects girls. And the other thing is that they have normal development until around a year and a half, or for the first 12 months, so 12 to a year and a half. And there's a regression and loss of skills from around a year and a half onwards. And the most common feature is like a hand wringing, so they kind of like go like that. It's also associated with learning disability, which is quite severe, profound, and yet almost exclusively affects females. Not to say that it hasn't affected males in some case studies, but the key points to remember for Rett is that they kind of appear normal in development till 12 to 18 months, mostly females. And it's really sad, actually. So yeah, children can live up to their early teens, but are usually wheelchair bound, and they have no language ability. So most of the problems that they have in Rett syndrome related to language. The next one is Lesch-Nyhan syndrome. So this is on XQ26 to 27, And the main thing I remember about this is they have kind of like a self-mutilation or like self-harming kind of behavior. So they can often headbang or bite themselves or kind of pick at their skin. And they also have some specific writhing movements. I actually think these are like some of the saddest disorders, to be honest. Like when I was doing these notes, I was like, it's really upsetting. The pathophysiology for this is an overproduction of uric acid, and that is because of this deficiency in hypoxanthine phosphoribose transferase, and that recycles purines. So if the question is related to purines, uric acid, self-mutilation, although that does come up in other syndromes, but self-injury, then it's probably Lesch-Nyhan, and it's also X-linked recessive as well. So that's Rett and Lesch-Nyhan. And then Angelman syndrome. So I think this one is quite interesting, the way that it's kind of got this nickname of happy puppet syndrome. And that's because they've got this kind of way of moving their arms. So when they're walking, their arms are kind of like, I think I watched a video of it on YouTube, so you can just Google that. I don't really, I think their arms are like kind of like upwards, so they've got like a flapping movement. And also they've got quite like a pleasant demeanor. So this is why it's kind of called happy puppets. They're kind of smiling and their arms are kind of flapping around, which sounds a bit bad really, but that's maybe a good way to remember it. They do have ataxia as well, verbal delay, but they can understand quite well. And then they also have severe to profound learning disability with seizures and sleeping problems. This is one of the genomic imprinting disorders, which again we'll mention later specifically, but this is 15q11 with maternal origin. I'm sure you've come up with this, what am I trying to say? I'm sure you've seen this in questions. It's kind of a very popular question in the question banks about maternal and paternal origin for the two syndromes, which are Angelman and Prader-Willi syndrome, which is the paternal 15q11. I just remember that as paternal and Prader, like just sounds like father, Prader kind of thing. So that's paternal. And the main things to note about that one is that that's a paternal origin disorder. They've got excessive weight gain from quite an early age, and they also eat without stopping. Basically, they have hyperphagia. They have a short stature, and they have a compulsive behavior, so skin picking and other kind of compulsive traits. Mild learning disability, small gonads, and hypotonia. So these two go together: the Angelman and the Prader-Willi syndrome. The next one is a Cri du Chat, which means cry of the cat, and that is because they make this characteristic sound of like a kitten, like making a noise, like a meowing, and that's a 5p deletion. They have hypotonia, hypertelorism, downturned mouth, and microcephaly. So yeah, I think they the main thing is just the sound that they make, which is characteristic. So this is DiGeorge syndrome. So there's kind of like a selection of clinical kind of features, I suppose you could say, which kind of go with this DiGeorge syndrome velocardiofacial, and it's kind of known as catch-22. That's also mnemonic, so it stands for cardiac defects, abnormal facies, cleft palate, hypocalcemia, and deletion on chromosome 22. But basically, with these, there's kind of the main things to note is on the 22q deletion, there's cleft palate, and there's also increased risk of psychosis and psychotic illnesses as well. And it's not fully understood what the mechanism for that is. I think we touched on it earlier that, it can be complex, but there was a lot of studies looking at certain genes in people with DiGeorge syndrome. So yeah, that's the main thing to note for them. And they don't have a very long life expectancy, I think. So then 17p11 is Smith-Magenis syndrome. This is the other one aside from Lesch-Nyhan, which does self-injury, but they also do this sad thing, which is self-hugging. I think it kind of like is a comfort thing, and they also have a hoarse voice. But you can see there's like quite characteristic facial features for Smith-Magenis, with that kind of like brow and the kind of the mouth as well. So that's probably the main thing to note: self-injury, self-hugging as well. And then Wolf-Hirschhorn, I think this girl's really cute, but basically they have this thing called Greek warrior helmet face, which is a bit mean, but basically if you look at their face, it's kind of like a gladiator helmet, because it's got this like very sharp, like down-turned kind of brows, and then like the mouth is kind of like downturned as well. So it's like that. It says downturned fish-like mouth. So it looks like a mask, like a gladiator mask, a warrior helmet face. And that's on 4p, and they also have cleft lip, microcephaly. Obviously, they have the developmental parts like intellectual disability and the growth delay, and also seizures as well. So it's quite a lot of things for that one. And then Williams syndrome. I find this one really interesting because they have like this elfin-like appearance apparently. So, and they're very cheerful and pleasant, and they're very articulate. So they speak really well, but it's usually about like quite superficial things. And they also have hyperacusis, so they can hear really, really well, too well, and they overreact to sounds. And I just think that's a really interesting like combination of things, like all of those things together. And I wonder why, like, they've, that those things have gone together. But it's a 7q11 deletion. It can be detected using fluorescent in situ hybridization, and this deletion tends to take place during the recombination phase of meiosis. So yeah, this is Williams. So Williams syndrome, just remember, elfin-like. They have LD, but they can speak quite well, but it's quite superficial. They hear too well, and it's an autosomal dominant. But most of the time it's not inherited, actually, it's sporadic. So the inherited part is a bit misleading. And then looking at Wilson's disease, again, this is also a genetic inherited condition, also known as hepatolenticular degeneration. So basically, I'm sure you've all seen that Kaiser-Fleischer ring that comes up through uni a lot, and in sort of other exams as well. But it's a disease of copper storage. You've got degeneration of the lenticular nucleus. The lenticular nucleus is the putamen and the globus pallidus. And you get characteristically these hepatitis, cirrhosis, all these other things, liver-related things mainly, as well as renal tubular acidosis, hemolysis, and blue nails. So the diagnosis is done by reduced serum ceruloplasmin and a reduced serum copper, which sounds weird, but it's because that 95% of plasma copper is carried by ceruloplasmin, and there's an increased 24-hour urinary copper excretion. Treatment is penicillamine, which chelates the iron and the copper, and then there's also trientine hydrochloride. So they chelate it and they take it out of the system. This one is von Hippel-Lindau syndrome. So this is an autosomal dominant condition which causes basically neoplasias, so tumours, like malignant tumours all over the body in various different parts of the body. It's an abnormality in the VHL gene, which is von Hippel-Lindau, on the short arm of chromosome 3. Features of it include retinal hemangiomas, vitreous hemorrhage. You've got renal cysts, extrarenal cysts, loads of tumours everywhere, basically, pheochromocytoma, cerebellar hemangiomas, and other tumours in various various parts of the body. So you can see in the picture, they're kind of all over the place, really. And then Rubinstein-Taybi syndrome. So again, these people who have this have quite a friendly disposition. There is a moderate learning disability. There's an unclear genetic etiology for this syndrome, but it's thought to be a 16p deletion. The patients with this are quite short, with microcephaly, so they have a very small head, and there's not a lot of information actually that I think is required really for the exam on these particular disorders, but they are important and relevant to note because they do come up sometimes, even these sort of less common ones. So just to remember, 16p for Rubinstein-Taybi, and sort of microcephaly and a friendly disposition with moderate LD. Coffin-Lowry syndrome. So this is an XP22 genetic mutation. They have a short stature, they have this short, broad nose, severe LD, and this slanting eyes appearance. So this is a boy with Coffin-Lowry syndrome, which I think looks quite sweet, really. Yeah, but just to remember, that's XP22. The Niemann-Pick. So this image is of the cherry red spot, which is on the macula, which is quite characteristic of Niemann-Pick disease. They have a 11p15 genetic mutation. There's two types, which is A and B, and they get abdominal swelling as well and feeding difficulties. And those are the main things. Yeah, the cherry red spot on the macula is quite characteristic. Next is tuberous sclerosis. So this is a neurocutaneous disorder. So it affects kind of the nervous system as well as the skin, and kind of the external organs as well. It's associated with ADHD, autism, depression, anxiety, and psychosis. It is quite heritable. It's got 95% penetrance, it's autosomal dominant, but there is various expressions. So sometimes people have quite a mild form, and other times people have quite a severe form of tuberous sclerosis. There's a girl that I know from uni whose parents, her mum was like positive for tuberous sclerosis but had no symptoms, and her auntie was also positive but had loads of seizures and lots of other things because of like these brain tumors. So it can have variable expression. So these aren't malignant tumors, these are hematomatous tumors, so they're kind of just more like space occupying, and it can look quite sort of big and bulky tumors basically, and this is due to a mutation in the tumor suppressor genes TSC1 and TSC2, and yeah, the main features kind of that are relevant for psychiatry are that you can get these tumors in the brain that can present with various psychiatric kind of disorders, illnesses, as well as like seizures and other space occupying lesion type clinical features as well. And yeah, so it's genetically heterogeneous, so there's linkage to 9q and 16p, but again, those are just the main genes, that tumor suppression genes. So these are some of the other features of tuberous sclerosis that are important to note. So the ash leaf spots, kind of like a leaf shape area of no skin color, depigmentation, the shagreen patches, which are roughened skin patches on the lumbar spine. I think they were pictured in the previous one. So yeah, if you can see picture C, that's a shagreen patch. Then the angiofibroma over the nose, that one is B, that kind of butterfly shape is characteristic for that in tuberous sclerosis. Subungual fibromata, developmental delay, LD, epilepsy, some things on the retina as well as kidneys, tumours in heart, lungs and brain. And I didn't even put all of the stuff, like these are just kind of some of the key ones, but they've got loads and loads of different features as well. So not to be confused with neurofibromatosis, this is a different type of disorder. There is some overlap actually, but they are quite different. There's NF1 and NF2. So with NF1, you get the cafe au lait spots, and these are kind of like these patches, these darkened spots. They get axillary and groin freckles, peripheral neurofibromas, and Lesch nodules, which are kind of like nodules in the iris. They also have scoliosis and pheochromocytoma. But it's quite bad if you look at Google images of neurofibromatosis, which probably wouldn't recommend doing, but the tumours are really, really bad. Like they are all over the body. Like this picture is very, very like, I guess this is actually a picture of NF2. So I think they have less kind of more on the skin. But basically the neurofibromatosis images, they have like tumors all over their body, like completely covered, and it looks quite it looks really like distressing, I suppose, for them. Kind of it's on their face and all that kind of thing. So I would say like it's worth remembering because it's quite rare, but it looks quite severe. And then yeah, so in NF2 you get the bilateral acoustic neuromas, you get more intracranial stuff like schwannomas, meningiomas, and ependiomas. So these are all the cells that you find in the brain. And there's a lot more kind of intracranial stuff with NF2, whereas with NF1 it's more like cutaneous stuff. But they're both autosomal dominant. NF2 is more rare. NF1 is on 17 and NF2 is on 22. And then we have McCune-Albright syndrome. So this is a disorder which affects bone, skin, and endocrine tissues. They have fibrous tissues in their bones, which has another name, which is another name for McCune-Albright syndrome. I can't remember how it's said, but it's like a very long name. And then it's caused by a random gene, and there's mosaicism in that as well. So kind of like it's not completely necessarily always the same presentation, depending on the kind of penetrance and complexity of the inheritance pattern. But the features are these cafe au lait spots. So you can see that in the picture, those kind of darkened spots. They've got short stature, and they also go into puberty earlier than expected as well. And genetics of Parkinson's disease. So actually most of the cases are idiopathic. There's not really been a specific gene found for Parkinson's specifically. They have implicated the Parkin gene and also SNCA on chromosome 4, which codes for alpha-synuclein, which, as you remember, kind of aggregates as a protein. But basically, just to remember, there's no specific gene for Parkinson's, and it can just be basically be. Okay, someone messaged me saying there's no sound. Can you guys hear me? Is everyone able to hear? Okay. Okay, cool. Okay, thank you. So it's fine now, that's good. Yeah, I don't know what's happening earlier. I was having a bit of problem with this kind of logging in with the thing, but I think it's okay. But yeah, just that's great. Just let me know if it happens again. So with the so we're on Parkinson's. Yeah, so other genetic contributors, there's lots of different DNA variants, and basically it's not really necessarily specifically based on any particular thing. And I've just put all of these into like a table just to remember the location of the disorders. So we'll just go through them. You could just make a note maybe of the most important ones. So we've been through pretty much all of these, but chromosome 1, you've got Alzheimer's, which is PSEN2, disc 1 for schizophrenia and bipolar, disc 2 is also schizophrenia, and then RGS4, that's briefly mentioned, but that interacts with neuregulin. Then on 2 is 2Q, which is for ASD, but as said, autism has lots of different complex inheritance. 3 does nothing. I'm sure it does do something in real life, but nothing that we need to know about for now. 4 is 4P for Huntington's, the CAG trinucleotide repeat, and also if you remember 4P, the Wolf-Hirschhorn, which is that Greek warrior helmet face. And then 5 is 5P terminal deletion, which is Cri du Chat. Then we've got 6. 6P does dysbinding, which is schizophrenia, and 6Q, which does some of the mood disorders. I've not come across PrEP really in any other kind of place apart from in this 6Q, so I wouldn't be too concerned about it unless you see it again in kind of other notes and you think that's important. I never came across that in anything else. 7Q, and then 7 again from microdeletion for Williams syndrome, which is the elfin-like features and the smiling sort of face. Then 8P for neuregulin, 9 for tuberous sclerosis type 1, 10 does nothing. Again, probably doesn't do nothing, but we're just saying that it does nothing. And then the 11P is for rapid cycling in bipolar, and also BDNF, so brain-derived neurotrophic factor. 12 is associated with trait neuroticism. I don't know how important that is, but chromosome 13 is a trisomy, which is Patau. Wilson's disease is from chromosome 13. And then on 13Q, you also get this other one, which is DAOA, which is for schizophrenia and bipolar affective. 14 is for PSEN1, which is the early onset Alzheimer's disease. And then 15 is important to remember for the paternal, maternal, the genomic imprinting microdeletions. So if you remember, Prader-Willi's from the dad, because P is like paternal, Prader-Willi, and then Angelman's is that upturned sort of and smiling angel type child, which is from the maternal side. Same chromosome. And then chromosome 16, you've got the ASD and tuberous type 2. Then 17 is familial FTD, frontotemporal dementia. Again, that has more complex genetic inheritance, but just important to note that. And then 17P for Smith-Magenis and 17Q for neurofibromatosis type 1. And the Smith-Magenis one is the hoarse voice, self-injury and self-hugging. Trisomy 18 is Edwards syndrome, which we'll cover as well in the kind of trisomy section. And then 19 is APOE, which is relevant for Alzheimer's and vascular dementia. And Notch 3 for CADASIL, which we'll cover as well. But these are basically like the vascular dementia type of things. And there's also associated with ASD. And then in chromosome 20, you've got the PRP, which is an inherited form of Creutzfeldt-Jakob disease. And lastly, we've got the chromosome 21, which is the amyloid precursor protein for Alzheimer's disease. And obviously, as you probably know, trisomy 21, which is Down syndrome. Then chromosome 22 does neurofibromatosis 2, DiGeorge syndrome, which you remember is a 22q deletion, also known as velocardiofacial syndrome or CATCH22. And then you've also got COMT on chromosome 22, which is relevant for schizophrenia, bipolar affective disorder, and basically any of the psychotic illnesses relate to COMT because it's sort of involved in the dopamine breakdown, so that is very relevant. And then on the XY, so on the sex chromosomes, you've got Fragile X, you've got the Lesch-Nyhan, which is related to uric acid and proteins, not proteins, sorry, purines, Turner's XO, which we'll cover later, and then Klinefelter's XXY. So those are all of the sex-related genetic disorders. So just to make a note of those. And then we'll talk a bit about the genetics of dementia. So we'll just start that and then we'll have a break in about 5-10 minutes and then continue. So in terms of dementia, this is very, very important to remember actually for the exam, because this usually does come up at some point, because the dementias are involved in psychopharmacology, in basic neurosciences, as well as in classification. So it's really important to remember all of the genetics of them. So for familial Alzheimer's, so this is early onset Alzheimer's. So this is in people who get their Alzheimer's earlier in life, which is unusual and very rare, as it says, is 1 to 6% of all of the cases. And actually some statistics say it's actually 0.1% of all Alzheimer's disease cases. And it is quite unusual to have a pure early onset familial Alzheimer's. So it's autosomal dominant, and the main thing to note is the PSEN1, which is on chromosome 14. So that's pre-senilin 1. And it kind of makes sense, like pre-senilin, pre-senile, so becoming old too early. And that is in 30 to 70% of cases of early onset. And then pre-senilin 2, that only does around 5%, so that's a bit less strong for that one. But then the amyloid precursor protein does account for around 10 to 15%. But again, there's kind of some crossover and like complexity to the genetic inheritance, as you can imagine. But yeah, the pre-senilins account for the majority of the early onset Alzheimer's disease. And it's also worth noting that there's increased risk of Alzheimer's disease in Down syndrome, which is because they inherit an extra copy of the APP, the amyloid precursor protein gene, on chromosome 21, and that's the pathophysiology for that. So yeah, so just remember, early onset familial Alzheimer's, pre-senilin 1, just think 1 is early, like 1 is before 2, so PSEN1 and early onset familial Alzheimer's. And then we go on to the late onset Alzheimer's. So there's only one gene that has been identified for the late onset Alzheimer's, which is very, very important. When this is talking about pure like Alzheimer's, I don't really know even how they can tell that it's pure. Like, I feel most of like kind of our teaching has said that it's usually mixed dementia. But anyway, so the main thing to note for the exam anyway is that there's APOE is really important for late onset Alzheimer's. So APOE helps to break down the amyloid plaques, and there's 3 types: APOE 2, 3, and 4, which we'll cover on the next slide. But basically, the E4 variant is the key risk factor, and that is on chromosome 19. And people who have people who are homozygous for E4 allele, so they have 2 of those, they have a really, really increased risk, 10 to 30 times increased risk of getting Alzheimer's. And then people who are heterozygous for APO4 are 3 times more likely to get Alzheimer's, but that's less than being homozygous for it. So it's called the risk factor gene, but even if someone inherits APOE4 allele, does not mean that they will definitely develop Alzheimer's. And some people with the allele never get the disease. And actually, some people who develop Alzheimer's do not have any APOE4 alleles at all. So it's just, if you have them, you're more likely to be at risk. It's obviously not the same as definitely getting it, but it's just important to remember that for late. So if you remember PSEN1 for early, APOE4 for late, and then these are the types of the genes for the APOE4, the APOE genes generally. So basically, yeah, just remember that APOE2 is protective, 3 is neutral, so it doesn't really do anything, and then APOE4 increases the risk. So 2 is protective, 3 is neutral, 4 increases the risk. And this is just kind of like a summary of where they are. So the amyloid precursor protein, that is on 21, chromosome 21. PSEN1, which we know is for early onset, is on 14. PSEN2, which is also for early onset but has less of a impact, like 5% or something, that's on chromosome 1. And then APOE, which is for your kind of normal Alzheimer's disease, that's on chromosome 19. So just remember those, because they always kind of come up in some way or another, and it's worth just noting that. And then CADASIL, this is the thing I mentioned earlier. So this is basically cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy. If you look at the scan, you can see some kind of like around the ventricles, there's these areas of white, and that kind of is a characteristic scan of someone with CADASIL. They've got these, it's basically like a vascular thing. It affects the small blood vessels of the brain in the white matter, and it causes migraines, and it causes like lots of little strokes, also big strokes, but they have multiple small strokes as well. And it kind of progresses to a vascular type of dementia. And it's related to Notch3, which is on chromosome 19. So that is worth remembering too. And then we have the dementia with Lewy bodies. So the main one for this is that SNCA. But having said that, most cases are sporadic, so they just happen randomly without any particular kind of genetic component necessarily. It's, again, complex inheritance like everything. But SNCA, which is autosomal dominant, and again, APOE4, it does increase the risk, but it doesn't directly cause. So in summary, for dementia with Lewy bodies, there's no particular real kind of genetic kind of component, I suppose, but you can remember SNCA for dementia with Lewy bodies. So we just kind of got about 5 minutes to 3. So should we just take like a maybe 10-minute break and then come back at 3. And again, if there's any questions, just drop them in the chat. The next thing we're going to go on to is just finish off the dementias, and then we'll go on to some more of the genetic stuff. So yeah, I'll see you guys at 3. Okay, hey guys, it's just gone 4 past on my laptop, so I can see most of you are back from the break, so let's continue. We are on the dementias, genetics of the dementias. So the next one is familial frontotemporal dementia. So actually, if we just look at frontotemporal dementia by itself, most of the time it's sporadic. So it comes sort of its own accord randomly through whatever genetic processes occur. So this can be various different genes. As you can see, most of it, 80%, is unknown. So even in the random versions of FTD, it's unknown. And then out of all of those, 25 to 45% is inherited, so familial frontotemporal dementia. And out of that, the most important mutation to note is on chromosome 9, which is a C9ORF72. And so this kind of gene mutation is really, really relevant for familial FTD. But also you can see that they've got progranulin, which is on chromosome 17, and also MAPT, which is tau proteins on chromosome 17 as well. So, oh no, my cat's trying to come in. So yeah, sorry. So basically, just remember that sporadic is most of the sporadic is most of the frontotemporal dementias, and then out of familial, to remember C9ORF72. So most of the time it happens randomly. Those are the genes, and then the other 25% of the time, or 45% of the time, it happens familially and C9ORF72. Now let's have a look at the trinucleotide repeat disorders. So we have mentioned some of them already, but basically what they are is an abnormal presence of expanded sequence of trinucleotide repeats. And one of the hallmark features of the trinucleotide repeat disorders is this thing called anticipation, where basically with time, like with successive generations, there's more and more repeats. And that results in an earlier age of onset of the disease and severity of the condition. And that concept is known as dynamic mutation. So basically, with time, it kind of gets earlier and earlier, which is weird and interesting, because you would think that then by now there wouldn't be many people who have got these. And I think actually the amount of people has gone down quite a lot. But I still find it strange that there's still quite a lot of people who have these diseases. So yeah, Huntington's disease, which is CAG. So children with parents who have the Huntington disease gene, like who are carriers of it, have 50% chance of inheriting the disease. It's autosomal dominant. It normally starts in midlife, but can occur as young as 2 or 3 years old, or as old as 80 years old as well. But usually it's sort of midlife, and there is kind of progressive symptoms towards death, really, for about 20 years. It's incurable and just gets worse and worse. And yeah, there's a degeneration of the GABAergic and cholinergic neurons in the basal ganglia and the striatum, and that's why you get that classic Huntington's chorea, which is the movements, and it's a defect in the Huntington gene, which is IT15 on chromosome 4. And some of the features of Huntington's include the dystonia, saccadic eye movements, personality changes, depression, apathy, irritability, and as I said, the chorea as well. And just a bit more on Huntington's, the CAG repeat should actually normally be less than 27 times, but in Huntington's it's more than that, and that relates, as we said, to the severity of the illness and the age that it starts. And with the kind of progressive generations, the anticipation, so with more successive kind of families, like keep going down, children of children of children, it gets worse. And you can also get juvenile Huntington's, which is the picture here, and under 20s can be affected by juvenile Huntington's disease. In terms of macroscopic findings, there's frontal atrophy, bilateral atrophy of the head of caudate and putamen, which we mentioned before, with the kind of decrease in the GABAergic and cholinergic neurons that causes chorea. So just the atrophy, just remember that there's atrophy of head of caudate and putamen. That's quite a common question, and that causes the chorea in Huntington's, and there's also enlarged ventricles as well. In the microscopic findings in Huntington's, you've got these neuronal losses in the striatum, and I think there was something I was going to mention about that. I can't find it, but again, also neuronal loss and gliosis in the cortex as well. And these inclusion bodies, which are pictured in the neurons of the cortex and the striatum in Huntington's. So another one of the trinucleotide repeat disorders, which is CGG. So if you remember, Huntington is CAG, C-A-G, and then Fragile X is CGG. I was just trying to think of a way that the way that I remember it. It kind of looks like egg, and X is like you know, eggs, ovaries, egg, CGG. Maybe not the best. If anyone thinks of something better, they can let me know. They have large ears, large testes. This is an X-linked dominant disorder. CGG repeat in the 5 untranslated region of the FMR1 gene. So the CGG repeats disrupt the synthesis of the Fragile X protein, which is FMRP, which is known to be essential for brain function and growth. And that gene is located at XQ27. And that's why it's classified as a X. So Fragile X is actually X-linked, but can't be classified as dominant or recessive because basically there's some complexity to the way that it is, the way that it mutates, because you can have a mutated FMR1 gene without kind of having Fragile X. So this is a trinucleotide repeat disorder, but there can be kind of features that are inherited that are similar to it from a different kind of way of, a different mode of transmission, essentially. With them, it's important to note the cluttered speech and elongated face, minimal eye contact, the large ears, and just remember the FMR1 and FMRP. And again, males are more severely affected because with females, there's another X, so that can kind of help. Whereas with males, that's the only X that is inherited, so that would be worse for them. And then myotonic dystrophy. So this trinucleotide repeat is CTG. This is inherited myopathy. The general features are they have this kind of long, haggard, kind of like messy appearance, it says, which is a bit mean, but they get frontal balding, bilateral ptosis, cataracts and dysarthria, myotonia, so the tonic spasm of muscle, weakness of arms and legs, and mild mental impairment, and diabetes mellitus. And there is a difference between the two. So with DM1, you get the, that's chromosome 19, and there's more distal weakness. So the weakness is kind of at the ends, the arm, like the bottom of the arms, bottom of the legs. Whereas in DM2, it's more proximal, so it's kind of pecs and more like chest and like higher up, like the closer, more proximal muscles as well. But both of these are inherited myopathies, part of myotonic dystrophy, and they both kind of affect a lot of different types of muscle. And just to remind you, so yeah, so DM1, more distal, chromosome 19, the DMPK gene, and then we've got chromosome 3 for DM2, which is more proximal weakness. The ataxias, and I think this picture is quite scary, actually. This is in Friedreich's ataxia, so you can see that scoliosis is really, really bad. And yeah, so with these are also some of the trinucleotide repeat disorders which cause ataxias, spinocerebellar ataxia, which is CAG, and Friedreich's, which is that picture, which is GAR, GAA and CAG. And some of the genetic terminology. So these are kind of things that like can be a bit difficult to remember exactly what they mean. I feel like they're the kind of things that you need to relearn every time. Like I'm sure we covered this in A-level and then in medical school, and basically we still have to cover it again for this exam. So Aneuploidy, which just means an abnormal number of chromosomes, so it can be more or less. It doesn't necessarily need to be either, it's just abnormal number. We have 23 pairs, total that's 46. The first 22 pairs are the autosomes, and then the 23rd pair is the sex chromosomes. So aneuploidy, which is the abnormal number of chromosomes, usually arises from a non-disjunction of the chromosomes in meiosis, which is when the sex cells are formed, like the gametes, and trisomies, which are when you have kind of 3 chromosomes, are usually errors of maternal meiosis 1. So trisomies, as you can see, there's 3 chromosomes there on that chromosome 13, so that would be Patau. These are the most common aneuploidy, so the most common, like having an abnormal number of chromosomes. They affect 0.3% of all live births. And usually autosomal trisomies are normally incompatible with life. So basically, the ones that are not the sex chromosomes, so like, any other chromosome like 12, 15, 19, when they have an abnormal number of chromosomes, they tend to abort spontaneously because they are incompatible with life on the whole. Whereas the ones that kind of do survive often are these ones, so 13, 18, 21, Patau-Edward and Down syndrome. And these sex chromosome aneuploidies are tolerated better, and they do tend to survive and almost have normal lives. We'll touch on that shortly, but yeah, the sex chromosome ones are tolerated a lot better in terms of being able to survive, having compatibility with life, have normal development. Yeah, so sex chromosome trisomies often are fairly benign. They may show sort of reduced sexual development and fertility, but they have normal lifespans, whereas those autosomal ones tend not to. And this likely relates to X inactivation and also the fact that the Y chromosome does not carry a lot of genes on it. So we'll go through those in a minute. So some of these other monosomies, so that means that mono, so they've got 1 chromosome. So 1 chromosome is missing and the total number is 45. And the only viable ones, the only one that survives in humans is 45XO. And yeah, Turner syndrome is less, we'll cover it again as well in more depth, Turner syndrome, but just to note that, yeah, that's the only viable monosomy that we have. And then again, the most common disorders that have an abnormal number of chromosomes are Down, Patau, Edwards, Klinefelter's, Jacobs, XXX, and Turner. So 4 of those are actually sex chromosome aneuploidies. So first of all is Down syndrome. So we know this syndrome very well. It's been studied in great depth. They have a short stature, simian crease, epicanthic folds on their eyes, brush-filled spots in their eyes, poor muscle tone, low set ears, often have mild to moderate learning disability. The risk is 1 in 800. Risk goes up with the maternal age, and it's trisomy 21. Patau is trisomy 13. Actually, Patau is really sad because I didn't even want to include a picture on the presentation because it's just really, really sad. The disorder, they die really soon after birth. They have really bad kind of malformations. They have no eyes. They have a cleft palate. Their kind of facial features are really like distorted, and it looks very sort of incompatible with life, to be honest. It's very, very sad. And they've got these fingers that overlap the thumb, which I put that picture in because something to remember it by. But yeah, very, very kind of unwell at birth because of these malformations. Yeah, so yeah, severe intellectual disability if they do survive longer than kind of a short time after birth. That is trisomy 13 Patau. And then Edwards syndrome, so this is trisomy 18. The children again die not long after birth. I actually saw a patient with Edwards syndrome when I did my SSC, like the student selected component with the air ambulance, and we went to this person's house, and there was a boy there who had Edwards syndrome, and he actually had a cardiac arrest, and then he died, and it was really sad, and it was really traumatic, and it put me off like peds and emergency medicine, because I was like, this is just too much. So yeah, it's really sad. They have these rocker bottom feet, which you can see in the picture. They're kind of like clubbed feet. They get like kidney malformations, very severe LD, and they are, again, quite unwell at birth and often don't survive for very long. Yeah, it's very sad. So less sad because people survive and have a normal life often, but equally a disorder of the genetic form is Klinefelter syndrome, which is XXY. These people live independently, can have a normal life, often go undiagnosed, unnoticed. They sometimes have a bit of intellectual LD with speech and language issues, and they often have some behavioural issues and can be a bit timid or unassertive. And they are tall with long legs and small testes. They do have a female type of pubic hair pattern and wide hips. So they have some kind of like more phenotypically feminine type of physiological features. But yeah, this is 1 in 1,000. So fairly common, really, actually. So they often, they live a normal life, but sometimes do have infertility in some cases. And interestingly, can sort of present looking fairly normal, having a normal life. So Jacobs syndrome, which is XYY, this is sort of known as the super males. They kind of used to say that these people were had more risk of being criminals and things like that, but that's not a thing now. People don't say that about them, because basically it's sort of thought that having XYY is linked as a socioeconomic kind of risk factor, rather than like a direct cause. They might have XYY, and then if they have socioeconomic risk factors that make them would already affect how they would behave, that kind of makes it worse. But without that, it's not like on its own a direct cause of being aggressive or having any other kind of problems. But they do have a very high stature, strong build, and normal fertility and testosterone. So they are quite sort of like considered manly, one might say. But they do have an increased risk of ASD and those emotional problems as well. So perhaps that may be why it was previously considered. This is a mosaic kind of type of thing. So there's a non-disjunction in the second meiotic division during spermatogenesis or post-zygotic mitosis. I wouldn't be concerned about learning that exact sentence, just to remember that they're the super male type and they're 47XYY. On average, they're 7 centimeters taller than most males, and they also have low set ears. So triple X syndrome, these again, people who have this live independently with a normal life. They may be slightly taller than average. They do have a global developmental delay and some LD, but they have normal sexual development and are fertile as well. And the rate is 1 in 1,000 in terms of incidence. Yeah, interesting. I find it strange that we don't come across kind of more people that have these disorders, because if you think about the population of, like, for example, of England or anywhere really, I would expect if it's 1 in 1,000 to see more. Well, I suppose you wouldn't see this, but some of these disorders, they seem to be more. They seem to have quite high incidence, but never come across them in day-to-day life. But again, I suppose it's because they're not very easy to detect, and why would you do genetic chromosomal analysis on any of these people if they had no symptoms? So Turner syndrome, so this is the only monosomy that is compatible with life in humans, which is 45XO. 1 X chromosome is partially missing or completely missing. Only occurs in females, and it occurs randomly is not necessarily like specifically linked to any genetic kind of candidate genes or anything. It varies very widely, but they usually have a short stature by age 5, a webbed neck, which you can see in the picture, widely spaced nipples, absent periods, and incomplete breast development. Most of them are infertile, but they can have assisted reproduction, and intelligence tends to be normal, but there is increased risk of learning disability, and they have difficulty with social skills as well. As Turner syndrome. So now we're going to have a look at the cell cycle, which is probably like, I feel, one of the less interesting parts of the syllabus content, but I suppose equally relevant if the Royal College says it is. This is a cell cycle, so it's got 5 phases. Well, technically it's kind of got 4 phases, but G0 is considered the resting phase when the cells stop dividing. Interphase kind of is all of the parts of the cell cycle apart from the mitosis part, and we'll go through it in order. So let's start from G1. So we're looking at G1, the cell, so it prepares the cell for DNA synthesis. Then in S, which is synthesis, DNA replication occurs. And then in G2, the cell is preparing for mitosis. So all of this is called the interphase. So purple, green, and pink. So that whole bit is basically preparing for cell division for mitosis. Then we move into mitosis itself. So as you see at the top, that's interphase. We've done that bit. The DNA has been replicated in the synthesis stage. We go into prophase, then we have a pro-metaphase, which is not in the picture actually, but is part of kind of metaphase. It's the bit before metaphase. Metaphase is the alignment of chromosomes in the middle of the cell. And mitosis is just creating, identical cells to the first cell. So this is not, this is just like generally like in all your body parts, this is happening all the time, and it's kind of like just copying the chromosomes each time, making identical cells. So metaphase, you've got the alignment of chromosomes in the middle, then anaphase, telophase, and cytokinesis when it splits into 2 daughter cells. It is just worth kind of remembering those because they come up in EMQs, unfortunately, but just to know what each part does is the most important bit, really. And I'd say that it's just something that maybe you just need to look at or watch a video or something. It's very hard to kind of learn from writing because it's a bit of a complicated kind of concept, but happy to go through it again at the end if anyone wants to. Then meiosis, this is how we create the gametes, basically. So these are genetically distinct from the parent cell compared to mitosis. So mitosis is basically just copying it, whereas this is like mixed up all the genetics kind of thing. It has half the number of chromosomes, obviously, because you want it to join with the other chromosomes from the other gamete, the egg or the sperm. And this has 2 different parts. It's got meiosis 1 and meiosis 2. So essentially, this is how they kind of, the body mixes up all the chromosomes so that when the child is created, I suppose that's why they have a kind of mixture of maternal and paternal features, like even from the same parent. So like some things they'll just be mixed up the way that it's kind of inherited. So 4 cells get created essentially instead of 2, which makes sense because you've got half the number of chromosomes in each. Centromeres are basically like a little belt in the middle of a chromosome. So where the chromatids join in the middle, they wear a belt, and that is a centromere that maintains the alignment in metaphase. And the position of the centromere divides the chromosome into 2 arms, long arms and short arms, and we'll just go through that. So as you can see, you've got the kind of P arm and the Q arm. So when you've got telecentric, there's like no P arms, it's just Q. Acrocentric is short P arms, long Q arms. And then sub-metacentric is short P arms, long Q arms, and then metacentric is kind of equal in both. And this is related to kind of the translocations and the way that when the cells are kind of dividing, there can be like it can just basically mess up and cause that belt to be in the wrong place, sort of thing. And those are called Robertsonian translocations. And mosaicism and chimeras. So the two different types. So the top in the picture, if we look at the picture, that's a mosaic. So you've got one zygote, so you've got one sperm and one egg come together, make a little embryo zygote thing, and there's one of the cells becomes like genetically mutated, if you see that blue cell, and then that causes kind of like mosaic pattern in the body. So like something wrong with one of the cells, that blue cell, that will, you know, go all over wherever, and that will come out phenotypically in the body in a certain way, but it's all from the same zygote. They're all from like one egg and one sperm. Whereas with chimeras, there's like two different genetically distinct population of cells. So there's more than one zygote. So you've got an egg and a sperm, and then another egg and another sperm, and those two kind of end up mixing together somehow, and that causes a chimera. Don't think that really happens in humans, to be honest. They've got a picture of a human though, so I don't really know. Maybe it does. As far as I know, it's only really been done in animal models for chimeras, but that is the main difference, whether it's one zygote or two zygotes, and genetically where they come from in terms of populations of cells. Okay, so nucleotides. So the basic unit of DNA is called a nucleotide, and it's these three bits. So there's pentose sugar, which is that little hexagon, which is either ribose or deoxyribose, depending if it's DNA or RNA. So deoxyribose is for DNA, ribose is for RNA. You've got a nitrogenous base, so that's one of the purines or pyrimidines. So the purines are adenine and guanine, and the pyrimidines are cytosine, thymine, and uracil. And the way I remember them is Ag for adenine and guanine, Ag, which is the symbol for silver. And I think silver is pure. I know it's a bit complicated to remember, but Ag is silver and silver is pure, like it's pure metal. So that's how I remember the purines, so A and G. And then the pyrimidines, I remember it like a triangle, like a pyramid, and the word cut, like C-U-T, and it's like cytosine, uracil, and thymine. So just remember cut, or you can think of like cut the pyramid, like cut a pyramid out of cheese or something. So those are the ways I remember the bases. You can think of your own ways, obviously, if you think that's very bizarre. It is quite bizarre. And then also there's a phosphate group as well on that nucleotide. So those three things make up the nucleotide. And then you get these codons. So three bases comprise a codon, and each codon codes for an amino acid. So basically there's 64 options because they come as threes. There are three that are stop codons, so that stops the kind of like, that's the end of the sequence, and that codes for a certain protein or codes for whatever. And the amino acids, so each of those three codons, not three codons, three parts that make up a codon form the amino acid. So for example, here you've got UUC, that codes for phenylalanine, and that's how that proteins are made. So lots of different codons stuck together. So copy number variations, I think we mentioned that earlier, but basically some portions of DNA can vary in their number. And yeah, so basically there's like copies of different areas of DNA, and that can cause more or less copies of certain genes. So in that picture, so that those genes can be just duplicated. Most of the time it doesn't have any effect clinically on any features or phenotype, but it has been noted to be associated with learning disability, schizophrenia, and autism, depending on what kind of area you're in terms of the copy number variation. Now we're going to have a look at some of the other inheritance type of terms. So heterogeneity. So what this means is that there's a presence of a variety of genetic factors which can cause the same condition or disease. So I know this picture looks a bit complicated, but if you just look on the left where it says A, you've got Cornelia de Lange syndrome, 1, 2, and 3. And then on the next bit, it's got kind of 1, 2, and 3. So basically, all it's saying in this diagram is that to cause this syndrome, you can either have a defect in 1, 2, or 3, and it will always cause the same thing. Whereas compared to cystic fibrosis, that always has to be the CFTR gene. So it doesn't matter like kind of where or what bit, as long as the CFTR gene is messed up, then that will cause cystic fibrosis. Whereas for this Cornelia de Lange, you could mess up different parts of the genes in different places and you could still get the same syndrome. So that's kind of heterogeneity. So that's presence of a variety of genetic factors or defects which cause the same condition or disease. And that is also important for psych because a lot of the diseases we look at do have heterogeneity. So for example, ADHD, autism, all of those, they're kind of what I was saying is complexly inherited. They have this heterogeneity, so you can get loads of different ways of inheriting ADHD. It's not one gene, it's the presence of a variety of genetic factors which can cause the same condition or disease. So I hope that makes sense. Then we're going on to Mendelian inheritance. So these are for single gene disorders. There's 5 different types. We'll just go through those. So and before we go through those, these are some of his laws as well, Mendel's. So he's got these laws which are, I think we did them in like A-level biology and haven't looked at them again since then. But basically, there's this law of segregation, which is that the 2 alleles for each gene are placed in different gametes. So in an egg and a sperm, you have like, obviously, say for example, chromosome 1, you've got a version of 1 in each in an egg and a sperm. So there's alleles for different things. So like in a man, there's like an allele for the eye color blue, and then in the woman, there's also an allele for eye color like brown. And the law of independent assortment. So the inheritance of one gene doesn't affect the inheritance of any other gene. So this means that basically they kind of don't influence each other necessarily. And yeah, I think that's just what he meant. Then law of dominance. So when 2 different alleles are present, only one is dominant and will be expressed. So for example, with the blue eyes and brown eyes thing, if one is dominant, so say brown eyes are dominant, and there's a blue allele and a brown allele, the brown one will be expressed. And going through his patterns, so you've got autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, Y-linked, and mitochondrial. So we'll just go through those and then show some of the pedigree charts as well. So with the autosomal dominant conditions, you can get it from like male to male, female to male, anything. It's a dominant gene. So if one parent is affected, there's a 50% chance of inheriting that mutated gene. Females and males affected equally. Parents are usually one affected heterozygote and one unaffected homozygote. And if one parent's affected, there's 50% chance of inheriting it, as I said. And then autosomal recessive, so male and females affected equally. There's 2 copies of the gene required for a person to be affected. And both parents are usually unaffected heterozygotes, so they usually don't like show it. The parents would not necessarily show the disease. And then 2 unaffected people who each carry 1 copy have a 25% chance with each pregnancy of having a child affected by the disorder. X-linked dominant, males and females both affected, but males are typically more affected than females because they only have 1 X, so they don't have another X to kind of compensate for that one. The sons of a man with an X-linked dominant disorder will all be unaffected because they will have inherited the X from their mother, which will compensate. And a woman with an X-linked dominant disorder has a 50% chance of having a fetus that is affected. An X-linked recessive, so males more frequently affected than females, is transmitted through carrier females to their sons in a Knight's move pattern, and affected males can't pass the condition on to their sons. a woman who's carrier of an X-linked recessive has 50% chance of having sons who are affected. I feel like this is really hard to kind of like understand in this kind of way, so maybe I'll just skip straight to the some of the examples. So some of the examples are here. So autosomal dominant, you know that, you know, if the parents are, one parent is affected and has a dominant allele, that will be expressed in the children in a kind of like a 50% chance. Recessive, it depends again. So with the key here, it says unaffected heterozygote. So they both carry a gene, the allele for a certain thing, but they may not display it because it's recessive. But then there's a 25% chance of that coming through, and then X-linked is kind of, you know, goes through the X and will can affect the females can be carriers, and it can affect the males in terms of actual phenotype. And then some of the other ones, so yeah, it's just it can just be a bit difficult to kind of formulate in your mind like how exactly it works. But I think when you go through it in question and are looking through, you can kind of understand how it would work. In terms of being recessive and dominant, those are quite straightforward, but then X-linked dominant and X-linked recessive, again, I think when practicing questions, that kind of helps to understand. So the main conditions for autosomal dominant are neurofibromatosis 1 and 2, tuberous sclerosis, achondroplasia, Huntington's, Noonan syndrome, and Marfan's syndrome. Then the autosomal recessive conditions are galactosemia, Tay-Sachs, Friedreich's ataxia, Wilson's, phenylketonuria, homocystinuria, Hurler's syndrome, and cystic fibrosis. These are all the autosomal recessive conditions that are relevant. And then X-linked dominant, mainly 2, which is Rett, and vitamin D resistant rickets. Then X-linked recessive, we've got cerebellar ataxia, Hunter's, Lesch-Nyhan, Duchenne muscular dystrophy, haemophilia A and B, red-green colour blindness are all X-linked recessive. So, yeah. Mitochondrial conditions. So mitochondria inherited from the maternal side. So these are always kind of inherited from the maternal, but they're mitochondrial. So we don't say X-linked because it's not from the chromosome, it's actually the mitochondria. So Kearns-Sayre syndrome and Leber's hereditary optic neuropathy are the two for the mitochondrial. Duchenne muscular dystrophy. So they've got something like they've got this calf muscle pseudohypertrophy, which is kind of known as a champagne bottle leg sign. This is an X-linked recessive disorder, mutation in the dystrophin gene in XP21. There's a frame shift mutation, so there's a shift in kind of like the codons, basically, like one like gets missed, so the whole like sequence becomes disordered. They get proximal muscle weakness, which is progressive from age 5, and what the symbol, kind of the picture showing, is that Gower's sign, where they kind of have such bad proximal muscle weakness, they use their hands to help them. They kind of push their hands up their legs to try and help them stand up, and there's LD in about a third of patients with this. And then Becker is kind of like the same thing, but less severe. Essentially, it's a more milder form of the mutation in the dystrophin gene. They still get these pseudohypertrophy of the muscles of the calves, but it develops after age 10, and there's less of LD. Again, it's X-linked recessive. So some more on inheritance. So looking at gene mapping. So basically, really found these concepts very difficult to understand. But mapping the genome basically means finding out which gene is responsible for doing what. That's all it means, really. And there's different ways of doing that. So you've got these kind of like genetic maps. So that's using the pedigree, like those Mendelian kind of charts, and looking at families and saying like, oh, this person's got that and a child got that, so that means that that gene does that. Whereas physical mapping is working out using kind of like the genetic parts of the oh no, someone said we're losing the sound. Can you guys still hear me? I don't know what's up with it today. Yeah, I'll repeat the gene mapping again. Sure. So I just said with the gene mapping that there's basically two ways to map the genome, and what mapping the genome means is working out which gene is responsible for doing what. So like what gene does I don't know, eye color. We do know that already, obviously, but basically working out which thing does what. So the genetic mapping is the pedigree analysis, which is the charts I showed you earlier, which were developed by Mendel. And basically you can work out genetics using that by saying like the father's got brown eyes and this son had brown eyes. So then working out what kind of genes does what. And then physical mapping is looking at DNA markers between DNA-based pairs. So actually looking at the understanding of the DNA structure itself, using different tools to kind of work out what gene does what. So these are kind of like, this is more like, I guess, molecular science level that we're looking at. And then the three types are chromosomal or cytogenetic mapping, radiation hybrid mapping, and sequence mapping. Okay. And then LOD score, so genes that are close in proximity on chromosome. So what essentially this means is that in meiosis, when some of the chromosomes are kind of being pulled apart to make that half number of chromosomes in each gamete, there are some genes that are unlikely to be separated because they're near to each other. So like physically on the thing, when they're pulled apart, like some of them won't be. Maybe the top and the bottom might be pulled apart because they're far away from each other, but the ones that are close together are kind of referred to as linked. And this LOD score basically determines if there's linkage between certain genes. So a LOD score more than 3 means that there is linkage, and then a LOD score less than -2 excludes linkage. And those are the main numbers for that. So for example, this is important because what it does is it kind of tells you what whether traits are linked. So, for example, say people with brown eyes usually have brown hair, or dark brown eyes usually have dark brown hair. So there is linkage between that because the genes for doing those kind of like coloring of the human's skin or hair or eyes are linked together, for example. So those would have like a very high LOD score of more than 3 because there would be linkage. And then, for example, maybe saying like people who are muscly and like inherit like a very big muscle build, and then maybe like length of fingers, that may be not necessarily correlated. So maybe that would have quite a low LOD score, -5 or something. I'm just making those up, but just to give you an example. And then concordance. So concordance is basically looking at the presence of the same trait or disease in two twins. And there's two main things to note from this. These are the only important things, really. So proband wise, this means that when you're looking at a twin study, when someone is conducting a twin study looking at twins in research, proband wise means that they're looking at the risk of getting a disease in each twin, like as an individual. That's more important, more meaningful, because the other way is looking at pairs of twins. So you're looking at the risk related to the pairs rather than the individual. So you're saying like the risk of a pair of twins inheriting ADHD is this much, which is useful obviously in research, but it's actually not that relevant and important for the person. So proband wise is more meaningful as it looks at the risk for each individual twin. Pair wise looks at the risk for a pair of twins, and obviously the pair of twins are not one person, so they're not, that's less relevant. That's the only real point about concordance that's relevant. So endophenotype. So this concept is basically it's interesting. It was introduced to psychiatry by Gottsman and Shields in 1973. So if we break it down and we say, like, we all know what genotype is, which is like your genotype is all your genes that you've inherited. And then we know what phenotype is. It's like the expression of those genes. So like, what do you actually look like? You know, what's your build like? What's your height like? That's your phenotype. And then endophenotype is like in between the two. So it's like, how does your genotype turn into your phenotype? So that's kind of what is understood by endophenotype. So these are measurable components unseen by the unaided eye along the pathway between disease and distal genotype. And endophenotype may be neurophysiological, biochemical, neuroanatomical, cognitive, lots of different things. And in terms of psychiatry, the way that's relevant to us is that in terms of psychiatric diagnoses, they can be kind of broken down into these things. So say you've got a gene that codes for like a certain, I think I wrote this example here. So yeah, so for example, a gene has a defect that blocks one specific biochemical compound, which is involved in attention. Say it blocks like some very like small little chemical that's like related to working memory or to attention or concentration. And then that translates into a behavioral endophenotype, which is like poor attention for working memory tasks. So it's not like a disease, they just find it harder to concentrate because they're lacking in this chemical. And then that may be clinically present as ADHD, as a phenotype. So when someone sees them and they say they can't concentrate, they've got poor working memory, all the stuff, then they say, oh, this person's got ADHD. But you know, not necessarily like ADHD, as we know, is not like a pure thing. It's kind of based on lots of complex genetic things. And this is what it's saying, that endophenotypes are the in-between parts from genotype, like whatever gene they've inherited, and the phenotype, which is how is that expressed in real life in the clinical aspect. So that's kind of what endophenotype does. So that pathway between genotype and disease. So the criteria for endophenotypes, so they must segregate with illness in the general population, they must be heritable, they must be state independent, they must segregate with the disorder within families, they must be present at a higher rate within affected families compared to general population, and they should be a characteristic that can be reliably measured and specific to a condition of interest. So I think it's a bit complicated to kind of understand really, but essentially these endophenotypes, this kind of, it has to be relevant to the expression at the end, but is not necessarily a specific genotype. Epigenetics is genetic involvement of things that are not necessarily like from the DNA itself. So like if you imagine, they talk about switching on and off genes, determining which proteins are transcribed. So, you know, for example, alpha-1 antitrypsin deficiency, like if people smoke, that can like trigger that to become, you know, activated almost. And that's sort of related to environment and lifestyle. And in this example here, it says like, Generation 1, a woman who smokes while pregnant induces epigenetic changes in 3 generations at once. So in herself, in her unborn daughter, and her daughter's reproductive cells, because all of the eggs in the ovary are made while they're inside the baby, while the baby's inside the mother. So that's affecting kind of 3 generations really. So you can see in that picture what's happened there. It's quite interesting really. Penetrance. So this is a probability or expression of a gene or trait. So like how likely is it that those genes will be expressed? So complete penetrance is like if the genes are present, all the population with them will have those genes expressed. And then incomplete is that genetic trait is expressed only in only part of the population that have the genes. So you can see the difference there in the green for the complete penetrance and incomplete penetrance. So individuals who have the same genotype for the gene of interest. Genomic imprinting we mentioned before because that's the thing that's relevant to Angelman and to Prader-Willi, which is the maternal and paternal inheritance aspect. So certain genes are expressed depending on the parent of origin, and DNA sequences are imprinted in the ovaries and testes, and these are specific for a mother or father that when they have a child, whichever thing is inherited from either. And most things are not specifically related to this. But the main examples are Angelman and Prader-Willi. So yeah, so we know that's 515Q for both. So segregation analysis versus linkage analysis. So these two terms sometimes come up. Segregation analysis uses family data to determine the mode of inheritance for a phenotype. So it's looking at like the pedigree, looking at the family history, looking at who's had what, and working out how it has been inherited, like recessive or X-linked or dominant. So this is using that information that you've gathered from how people have presented with a phenotype, so their clinical expression. Whereas linkage analysis, so this looks at the genetic location of a disease gene that is inherited by all family members that have the disease. So this is looking at more like the molecular level, the genetic location of a disease gene. So that's the main thing. I feel like the terminology makes it more complicated than the actual concept. So lionization. So for this, I think of lionesses because they're the female lions. Basically, the females silence one of their two X's, and the inactivated X chromosome is condensed into a bar body, which is just that kind of little lump there. And this is a random process. So basically, it occurs in all somatic cells of women, but not in the ones that form the gametes. So X inactivation occurs in most, but not all genes on the X chromosome. And if a cell has more than two X's, then the extra X's are also inactivated. So yeah, it's just inactivation of the X chromosome because we only need two to work, obviously, as you know. Heritability. So what does heritability actually mean? What it really means is the proportion of phenotypic variance which is attributable to genetic variance. So if you ask yourself this question, what is the proportion of a variation in a given trait within a population that is not explained by the environment or random chance? So it's kind of like the main point of this for the exam, I guess, is saying that it's not about heritability of like an individual, it's looking at the population. So you're looking at what are the genetic factors that cause this thing. So it says here that if a condition has a heritability of 0.7, then 70% of the variance in that condition seen in the population is attributable to genetic variation, and the 30% is due to environment. So I hope that makes sense. So in terms of proportion, the range is, like, say the number is 0.0, so genes do not contribute at all to the phenotypic individual difference, and then 1.0, so genes are the only reason. And for human behavior, almost all estimates of heritability are in the moderate range of 0.3 to 4.6. So like, for example, like Huntington's disease, that would have a very, very high heritability because almost all of it in the population would be related to genes, essentially. So what you're basically saying is how much of this is likely to be related to the genes rather than environment or random chance. Then some other terminology, we've got splicing. So when DNA is transcribed to the mature RNA, introns are removed and the remaining parts, exons, are translated. So this is the other way around from the way I think about it. You'd think that exons are removed, but actually introns are removed. Exons are kept. So I always just think that it's the other way around from what I think it is. Then the spliced form of RNA, so when you've taken out all the introns, is called mature RNA. And then proteasome is that's the thing that degrades proteins inside the cell. And then the recombination fraction is a measure of the distance between loci on a gene. Penetrance is the proportion of the people with mutation who manifest clinical symptoms. Multifactorial inheritance, we mentioned before, was conditions resulting in interactions from parents and the environment, so lots of different factors causing inheritance. Haplotype is a set of DNA variations that are often inherited together, so like brown eyes, brown hair. Karyotype is just one person's individual collection of their own chromosomes. That's just their karyotype. And then locus heterogeneity, which is a disorder. A disorder is caused by mutations in genes at different chromosomal loci. So I think we mentioned that with the different examples. So with that one where it said that the opposite of the cystic fibrosis one, the other one where it had lots of different options of how the mutations could cause a disorder. So now we'll just quickly talk about the Hardy-Weinberg principle. We've just got a few slides left, so we're just coming up to 4 now, so it should be finishing up soon. Hardy-Weinberg, so basically these are the 5 conditions for all the kind of genes and frequencies of a population to remain constant. So it's like a gene pool, so like imagine a little island of people. They only all the whole population stay on this one island, and that gene pool is going to remain constant because it's in equilibrium, because basically you're not having migration of people, there's kind of random mating, and essentially you're kind of just mixing everything up, and there's not any kind of changes in it. That also says in the law that they can't have any mutations or any natural selection either, and the population must be large enough for it to remain random as well. And then in terms of the actual equation itself, it's p squared plus 2pq plus q squared equals 1. I think maybe just learn it as is. That does sometimes come up as like work out the frequency of whatnot, but I think if you know the formula, then you can just work it out. And p is frequency of dominant allele, and then the q is the frequency of the recessive allele. So yeah, so these are just the frequency of the homozygous dominant, the frequency of the heterozygous, and the frequency of the homozygous recessive. So what you would normally find in a population, and then how that kind of comes out as an equation. The molecular biology techniques, so there's kind of 4 that are relevant. Southern blotting detects DNA, northern blotting detects RNA, western blotting detects proteins and uses gel electrophoresis. And a common question is, which technique does the confirmatory HIV test? And it's western blotting. Whereas which technique does the initial HIV test? This is ELISA, and that detects antigens and antibodies. So that's probably the only relevant part of that. Although I think I did have a question in December for blotting, so worth knowing that too. Types of mutation, and this is my last slide. So these are mutations in the kind of long DNA sequence, which is made up of codons, which are made up of amino acids. So if you have, like, imagine a long string of like A, G, you know, CAG, GCC, you know, all of those kind of things going along as a sequence, and they kind of split into 3, making the codons, which code for the proteins, code for the amino acids, and that codes for the proteins. If you can imagine, if you take one of the letters out, that will cause a mutation in the amino acid, and then that will change the protein as well. And these are the types of mutation that you can have. So silent, so the mutation has changed. So one of the amino acids has changed, but one of the codes has changed, but it still ends up being the same amino acid. So say CAG and CGG turn out like that, makes the same thing. Point mutation, so that's changing one base of gene sequence. Frame shift is like one is taken out or one is put in, it just shifts the whole thing along. And then nonsense mutation means that a stop codon is formed. So there's 3 types of stop codon. And then a missense mutation, it means it alters the codon and it causes a different amino acid to be made. And that is it. So we've just come up to 2 hours, and now I've got some time for questions. As usual, I will hang around till all the questions are done, or if there's anyone who wants to ask anything specifically, or about the exam, or just wants to have a chat. So yeah, any questions, just put them through on the private chat and I will answer them or go through any of the stuff again if you would like. Otherwise, thanks for listening, and I hope that was useful for you. So one of the questions is about the great, thank you. I'm glad you guys enjoyed it. Getting some nice messages. Thank you so much. Yeah, one of the questions is about the aneuploidy. It's just either it can be more or less chromosomes. The question is about what it kind of means. It can basically be any of the chromosomes just has an abnormal number. So like if you look in this picture, you've got 3 there in the trisomy. But then say if you had like the 45XO, that means 1 is taken away. So you've got 1, that's also aneuploidy. So both of those are considered aneuploidies. Yes, that's it. Okay, I feel like it was a very heavy session, to be fair. We did have 100 and it was like 113 slides for today, which is probably the most. I think it's because all of these kind of syndromes are just they're really relevant and really important, but I know it's quite a lot, so don't panic. But this is pretty much everything you do need to know about molecular genetics. If you do cover all this stuff, you're kind of done. Good. I'm really happy. People are happy. Cool. Okay, yeah, so I can't see any more questions that are related to this topic, so I will just hang around for another sort of 5-10 minutes, and then if there's not anything else, then obviously see you guys next week, and hope that you get lots of work done this week. You guys are going to smash it. Just keep going. Not very long left at all. Do your mocks, do your question banks, and you just do it. You're doing great. Just smash it out, and yeah, just smash it out, and you'll be fine. It's just the last final bit now. So, as always, feel free just to drop me a line if you want to talk about anything in particular or any queries about, like, obviously, as you know, I've said I'm not an absolute expert on like how to revise, and everyone's really different, but yeah, if you want any advice on specific things, how to revise specific areas, or you know what exactly resources to use, and do just drop me a message. Happy to help. Otherwise, yeah, just enjoy, smash it, do your thing, be happy. Yeah, thanks, guys. It's really nice to hear that it's helping and that you guys are benefiting, obviously. Yeah, no, I'm really happy, and thank you for your attention and involvement. So, looks like most of the questions are pretty much done, to be honest. So, I'm going to end the webinar. If anything else comes up, just drop me a line. But otherwise, I'll see you guys next week for the next session, the psychopharmacology. Same time. We've got 3 sessions left. So we've got 2 on psychopharmacology, and then we've got 1 on classification, and that actually brings us up to the exam date. So hopefully by the end of this, you will already be kind of doing all your own stuff as well in terms of questions, and hopefully we'll get to the end of this together and you'll be smashing it. So speak to you guys soon. See you next week. Have a great week ahead and keep working hard.